This video will demonstrate the steps required to do the volumetric analysis of the mass of vitamin C in an over-the-counter vitamin C tablet. Our goal is to use this analysis to assess the accuracy of a reported value of vitamin C in the tablet. This kind of analysis is essential in the real-world context, where quality control of the active ingredients in drugs is vital for public safety and market reliability. The steps described in this laboratory procedure build on the steps conducted in Lab D, which was the standardization of the secondary standard, sodium thiosulfate. This diagram provides an overview of the steps in this analysis. A single vitamin C tablet is dissolved in water, and this solution is made up to a final volume of 100 ml of solution. A known amount of aqueous iodine is generated in an Erlenmeyer flask by combining a limiting amount of potassium iodate with an excess of potassium iodide and an excess of sulfuric acid according to reaction 1. Some of this aqueous iodine is consumed by the reaction with the vitamin C contained in a 1 ml portion of the vitamin C solution, according to reaction 2. The amount of aqueous iodine remaining after the reaction with vitamin C can then be determined through the titration with the standardized sodium thiosulfate solution, according to reaction 3. This volumetric approach is called a back titration, as it involves an indirect determination of the amount of analyte, which in this case is the vitamin C. The first step involves dissolving the vitamin C tablet in water. We are using a tablet from the Jamieson brand, which is reported to contain 500 milligrams of vitamin C. While the vitamin C, also known as ascorbic acid, is very soluble in water, the tablet contains filler, some of which is not very soluble in water. Here we see Scott record the relevant details about the vitamin C tablet and add the tablet to a 100 ml beaker and then add about 70 ml of deionized water. We will go on to other steps in the procedure while the tablet dissolves. In Lab D, a concentrated standard solution of potassium iodate was prepared. This now needs to be diluted tenfold by pipetting 10 ml of the solution into a new 100 ml volumetric flask and diluting to the 100 ml mark with deionized water. In the lab, we see Scott demonstrate the correct protocol by rinsing a dispensing beaker and then a 10 ml pipette with the concentrated potassium iodate solution. Scott also rinses the 100 ml volumetric flask several times with DI water to ensure that this flask is clean. He then uses the pipette to precisely transfer 10 ml of the concentrated solution to a new 100 ml volumetric flask. Enough deionized water is added to bring the liquid level to the designated mark. Note the added precaution of removing any water from the upper part of the volumetric flask before thoroughly mixing. In the next step, the burette is prepared by rinsing it with standardized sodium thiosulfate solution from Lab D. The burette is then filled with standardized sodium thiosulfate solution. At this stage, we return to our vitamin C solution. The vitamin C has dissolved, but the solution is cloudy due to the insoluble filler. Filtering through glass wool will help remove some of this filler. It is worth noting, however, that although the filtration step is shown here, we have also found that students obtain accurate results without the filtration step. Enough deionized water is added to bring the liquid level to the designated mark. Note again the added precaution of removing any water from the upper part of the volumetric flask before thoroughly mixing. We are now finally ready to prepare a 125 ml Erlenmeyer flask with the reaction mixture to produce aqueous iodine, which will then be reacted with a portion of the vitamin C solution. To ensure that a sufficient amount of iodine is produced, we need to increase the volumes of dilute iodate and sulfuric acid solutions in comparison to what was added in Lab D. Therefore, we pipette 15 mL of the dilute iodate solution instead of 10 mL, and we add 10 drops of 1 molar sulfuric acid instead of 5 drops. A mass of 0.1 grams of potassium iodide will be sufficient of an excess of iodide ion. Scott rinses an Erlenmeyer flask with deionized water. He then uses a pipette, which has been previously rinsed with the dilute iodate solution, to quantitatively transfer 15 mL of this solution. After adding 0.1 gram of potassium iodide and 10 drops of 1 molar sulfuric acid, the solution turns to the characteristic orange-brown color, which indicates the presence of iodine. While it's difficult to pick out in a video presentation, there is a slight fading of the orange-brown color when Scott pipettes the 1 ml of the vitamin C solution into the Erlenmeyer. This is, of course, due to the fact that the vitamin C reacts with the iodine. Prior to starting the titration, Scott rinses the sides of the Erlenmeyer to ensure that all the reagents are combined. The stages in the titration are the same as what was experienced in Lab D. The thiosulfate titrant is added until a faint yellow color is observed. This indicates that most of the aqueous iodine has reacted with the thiosulfate anion. Because it is difficult to observe the disappearance of a faint yellow color, about 2 to 3 mL of starch indicator is added. The starch combines with the remaining aqueous iodine to produce a dark blue-purple color. The titrant is then added to the point that the blue-purple color completely disappears. Scott reads the initial volume of the burette to 2 decimal places. A white sheet of paper below the Erlenmeyer flask allows for better visualization of the color changes. Scott demonstrates proper technique by cupping and controlling the stopcock with one hand and swirling the Erlenmeyer flask with the other. The solution arrives at a faint yellow color. This squirt bottle dispenses about 2 to 3 mL of the starch solution. Scott also takes a minute to wash down the sides of the Erlenmeyer flask to ensure that all drops of titrant have contacted the analyte. One final drop turns the solution colorless. The final volume of the burette is recorded to 2 decimal places. The titration is repeated 3 more times for a total of 4 titrations, so that 3 of the titration volumes are within 0.25 mL. After watching this video, a complete set of titration data will be provided through a link in Moodle.